Everyone. Welcome to the Department of Geriatric Medicine Grand Rounds. This CME activity is jointly provided by the Queen's Medical Center and the UH Jabson Department of Geriatric Medicine. Before I introduce our speaker, I have a few housekeeping items to go through. At this time, please enter the name and credentials of everyone viewing Grand Rounds for CME attendance purposes. As a reminder, evaluations are required for CME. You can find evaluations by following the link in the chat at the end of this talk, or in our email advertisement or on our website. Our sponsor will send out CMEs in mid-January every year. Your comments are very important to the planning committee and will be used to plan future programs. It's my great pleasure to introduce Dr. Kaz Nakagawa, who is Division Chief of Neurology and Professor of Medicine at the John A. Burns School of Medicine. He's also the chief of the Neuroscience Institute at the Queen's Medical Center, which oversees the inpatient and outpatient neurology, neurosurgery, and neurointerventional surgery divisions. He obtained his medical degree from the University of Florida, completed neurology residency at Mass General Brigham, and completed neurocritical care fellowship at UCSF. He practices as a neurointensivist and stroke neurologist, caring for critically ill patients with severe neurological conditions, including stroke and traumatic brain injury. More recently, he launched a separate organization called the Brain Health Applied Research Institute. His research endeavors involve understanding the clinical, genetic, and social factors that impact stroke-related health disparities observed among Native Hawaiians, and understanding how a longevity-associated FOXO3 gene mitigates the deleterious effects of hypertension on the cerebral blood vessels, potentially reducing the risk of ischemic and hemorrhagic stroke in the Kuakini Honolulu Heart Program study. Please join me in welcoming Dr. Kaz Nakagawa.
Thanks, Kamal. Thanks, everyone, for inviting me. I'm really excited to give this talk to you all. So the name of the talk is Neuroplasticity and Brain Health. These are my disclosures. So neuroplasticity. So what is neuroplasticity? It's our ability for the brain to undergo both structural and functional changes to form new network, quote unquote, brain rewiring. And this can happen. And this is a picture that shows a proof of concept that the couple neurons are sprouting their dendrites and forming stronger and more enriched connections. And this happened over 2 weeks and 2 months after stimulation. And this stimulation is not some fancy zapping the brain or technology or some kind of a high level medication. This was like a really like a brain exercise activity, brain puzzle that can do something like this. So it's very powerful. The structural neuroplasticity is really structural, so forming new brain cells, new neurons, and so on. And that is more common in the pediatric population, children, but it can actually happen in adults as well. Less prevalent, but what's happening more in adult population is the functional neuroplasticity. So even though we may not grow more and more neurons after we lose them, we can actually form more dendrites. So dendrites are all these sort of branches and tentacles that you see out of the cell body, and they can actually sprout and touch with each other and form new connections. And that's what happens in adults. So after brain injury, stroke, or cardiac arrest, when some brain cells die out, some of the existing neurons can actually sprout out and form new connections and restore some of the functions. So again, this is sort of a simplified picture of what happens when there's a traumatic brain injury, stroke, cardiac arrest, whatnot, and then lose one brain cell. Initially, the function is lost. The neuron D, which you see on the bottom right, is no longer firing because neuron B was lost. But as the neuron A sprouts out and then reaches out to and give a little fist pump to neuron D, it starts to work again. So that's the concept. And this doesn't have to happen after damage or injury. It can actually happen in healthy adults as well. So the more we actually stimulate neuroplasticity, we can actually form tighter network. In fact, this is the reason why we can still learn we can still learn to play instruments, even if you never played before. It may be harder than when you're a teenager, but we still can. You can still pick up new sport. You can still learn to surf. It's all neuroplasticity. In fact, I was away in Australia a couple of weeks ago on vacation and had to drive on the right-hand side, completely opposite, all awkward. Took me a day or two to get used to it, and I kept telling myself neuroplasticity, neuroplasticity, neuroplasticity. And you know what? After a couple of days, I was able to drive just fine. So this is a little bit of a complex sort of figure, but this is kind of what it takes to promote neuroplasticity at our level. We don't have to go to tertiary quaternary centers to get some kind of a fancy intervention to do this. We can do it at home. So it takes new, challenging brain activity, whether it's new thoughts, problem solving, learn to do something action motor, whatever that is, novel. It has to be meaningful. You can't just go through the motion. It has to be a very strong activation of that pathway, where if you make a mistake, you're going to have a meaningful whoops instead of just a weak whoops, I made a mistake. It has to be like a meaningful action where you make a mistake, you want to correct it. And then it has to have an emotional component to it. With a meaningfulness, it creates a lot of emotional sort of triggered norepinephrine and so on. That's going to also help with the neuroplasticity process. Now you go through that motion, but you also need a good foundation to sort of lock that in, which is increased by exercise, diet, and sleep. Again, these are three things that we often say as a physician for our patients. Like exercise every day, walk every day, eat better food, get some sleep every day. But we have to have a better understanding of how that's going to improve brain health. And now, hopefully, I'm going to show you some of the science behind it. And lastly, my personal goal is to add art and music to that mix. My aspirational goal is for most of the primary care physicians to say, walk every day, eat well, sleep well, and do some art, 20 minutes of art a day. And I think art is also underutilized, less frequently talked about, easy to do at home intervention. Now, I'm going to show this picture of a fertilizer. You know, um, if you want the plants and crops to grow and sprout and be robust, fertilizer helps. Without the fertilizer, you can see the picture, no fertilizer with a fertilizer. Now, brain is the same thing too. The brain needs a brain fertilizer. So what is that? What is this brain fertilizer? It is BDNF, or brain-derived neurotrophic factor. And I just call it brain fertilizer. Some people call it brain steroid. But with this, it's going to boost the ability for the brain to do the neuroplasticity and makes a stronger pathway, stronger network. Okay. This BDNF helps with neurogenesis, so creation of new neurons in the hippocampus, which is important for short-term memory formation, and dendritogenesis, so it's the dendrites, the tentacles, the branches that pointed out. That's going to be more sprouting out, which is going to increase the likelihood for other neurons, dendrites, to be touching and forming new connection. And it's going to also strengthen the synapses, so synaptogenesis, so gene transcription to create new exchange of neurotransmitter and synapse, and then form a new connection. Now, in an animal model, you can do a BDNF gene transfer so that one animal can have more BDNF expressed, and it alters the architecture of the neurons and creates this halo of new basal dendrites. Those are the baby dendrites. So the key is the word alters. So by definition, alteration is a change. Change means plasticity. So again, the brain neurons can change. They can be altered. They're plastic. Okay, I just want people to understand that it can change. So, compared to the control group where you just basically do the same kind of thing, but do not transfer the BDNF, it doesn't change that much. And compared to the one on the right, there's a lot more expression of the dendrites and so on. Now, it's kind of hard to appreciate visually. I'll be honest. I was looking at them and I kind of about that. So maybe objectively, this tells the picture. Uh, this tells the story better. That when you have a BDNF transfer to the mice or something, that's the square one. And basically, you have more dendritic intersections away from the cell body compared to the control. And again, you have a lot more intersection of dendrites if you have the BDNF overexpressed. So, may wonder, what does that mean? Do I need to like get a gene transfer of the BDNF or something? Like you got to ingest some crazy medication? No, there are much more simple things you can do. So first, let's talk about what actually reduces your BDNF. Well, unfortunately, aging process. As we get older, we have just less BDNF. That's just the reality. If you have Alzheimer's disease, again, less BDNF, and if you have chronic stress, and that's something that's modifiable. That's something that we can work on. So what can promote BDNF? Exercise, enriched environment, and antidepressants as well. Just want to really focus on exercise. I'm not here to endorse antidepressants on everyone. I just want to focus on exercise because this is something that we can do. Again, my goal is for you to go back to your practice and endorse exercise with a more scientific sort of understanding of what it does to the brain. So how does exercise increase BDNF in the brain? Well, there are three major sort of pathways or mechanisms. One is a neuronal, it increases a neuronal activity. And the second one is a cerebral blood flow gets activated. And then the third one is you have a lot more exerkines from the peripheral tissues. So I'm going to go through one by one. So neuronal mechanism. So when we're really out there doing stuff, playing sports, surfing, running, whatnot, you have a lot of like neurotransmitters that are being secreted. You have a lot of serotonin, norepinephrine, dopamine, things that are exciting. And then these neurotransmitters actually, through epigenetic sort of phenomenon, can trigger more expression of BDNF coming from the brain. Again, brain-derived factor. It actually promotes more BDNF expression. And when we're exercising, increased cardiac output, more blood flow to the body and to the brain, you have vasodilation, a lot more cerebral blood flow to the brain. And that actually triggers the nitric oxide pathway, which also affects and promote BDNF expression. So there's a blood flow augmentation that leads to nitric oxide pathway and then BDNF activation. The third mechanism is through our body. So when we're exercising, our liver and muscles secrete hormones, factors, all these things that can actually also go back into the brain, process the blood-brain barrier, and increase BDNF formation. And one of them that I'm going to focus on is this, irisin. So you see that word irisin that I boxed. This is a very interesting hormones or neurohormones that's made by the muscle. So when you exercise, our skeletal muscle creates irisin, and which has three big merits, starting from the bottom, which is it has a positive benefit on coronary artery disease, hypertension, and heart failure. Okay. And then second thing is on the blood vessels. So irisin makes the blood vessels healthier. And lastly, it crosses the blood-brain barrier and it has a positive benefit on stroke, Parkinson's disease, depression, and ultimately increases neuroplasticity and your cognition. So this is the diagram for irisin and vascular health. I'm not a basic scientist, so I don't quite understand the exact mechanism, but through multiple scientific literature, it has shown to decrease blood pressure, also improves the atherosclerosis, and also it helps with a heart failure condition. What's interesting is that irisin more recently was discovered to clear amyloid plaque. The amyloid plaque that's associated with the Alzheimer's disease. Again, irisin is good for the BDNF, the brain fertilizer. Yes, it's good for like learning, but it also helps clear the amyloid plaque. Think about it. We focus so much attention on the end stage and giving the Leqembi and so on. here we are early in the stage, we can keep clearing the amyloid. So how does that work? Well, it actually works on neprilysin, which actually is the actual sort of protein that helps cleave and clear the amyloid. And this is the paper that I got the figure, which shows that when the irisin here binds here, okay, it activates, actually inhibits this, which inhibits through negative, double negative sort of mechanism. It creates more and more NEP, which is neprilysin, which helps degrade the amyloid and cleave it and then get rid of it. Okay, so now we can truly say that, if you really want to clear your amyloid early stage and prevent dementia, start exercising. Okay, so this is like I had an aha moment when I found this out. Okay, so to summarize this concept of neuroplasticity and brain health and exercise, aerobic exercise, don't ask me, high intensity and low intensity, but overall enhances the neuroplasticity by augmenting the brain-derived neurotrophic factor, which is BDNF, which I call it brain fertilizer. And also it primes the brain for learning and skill acquisition. So I say, if you want to learn something, go exercise early in the morning and then go to school and go to your job. It helps clear the amyloid plaque and helps lower the blood pressure. We have some stroke audience here, so I kind of put this together. There's a paper that was published by the, I think stroke, by the American Heart Association that looked at this. For the stroke survivors, the BDNF was also increased after high intensity aerobic exercise, but there was no increase when they were just in the regular sort of conventional physiotherapy, balance exercise, flexibility, and so on. So that's just to keep in mind. And I know for this audience, for elderly people, it may be somewhat challenging to do intensity aerobic exercise, but I think we can be creative. Now, really the goal is this, we all gonna have a brain function decline over time as we age. But what we want to do is change the trajectory so that by the time we die, our brain is still relatively healthy. And I think we can change this angle early on in life, in mid-age, like middle age, and then hopefully we're not going to be in the red line. So again, we often sort of see patients towards the end, but I think if you're interacting with people early in life, like 30s, 40s, and 50s, I think we should really promote exercise. Next slide. Next, I just want to change gear and talk about art. So neuroplasticity and art. So can art improve brain function? I gave a little pause because I just want you to stare at those two figures, to art. And I don't know if you noticed, but something changed a little bit. It's colorful, it's captivating, and it's kind of like draws your attention. And then perhaps for like a split second or two, you kind of forgot about work, maybe you kind of got into a different state of mind, somewhat of a Zen state. And that's the kind of impact that the art has on your brain. The functional MRI studies have shown that doing simple things like doodling, coloring, drawing can activate the prefrontal cortex and other parts of the brain. And then it can actually increase the blood flow to the brain. And also, it's been shown that doodlers are more analytical, their brain function better, and they can focus better. There has been a lot more attention on simple things like doodling and brain health. And when we're doing creative work like art or anything that's creative, we activate our default mode network, DMN, which is a resting state network. So that's when we're not doing anything, like idling state, which is really, really hard to achieve these days. We wake up and we start the day, we're constantly working and thinking about things. We barely have time to have like 1 minute where we can go blank. And that sort of having that blank moment is really important. It's even harder nowadays because we're getting constant text and tiger text and pages and everything coming at you. So this deformal network is really important for sort of a, I don't know, like basis for self-reference, checking in your own emotional state, being empathetic, thinking of other people, other people's feeling, moral reasoning, and then sort of like reflecting on yourself. It's just a reflection sort of state. And I can say go meditate, but it's hard to do that. And perhaps it's easier to do coloring, drawing 10 minutes a day. So, I've been personally telling my patients, and when I see a couple patients in the clinic occasionally, I prescribe doing daily art. It's easier to say, get a coloring book and then do art a day. And in fact, I want to show you, my family got me a coloring book, Star Wars coloring book, and coloring pencil for Christmas last year for this purpose. And I keep it in my office and do about 10 minutes of that, hopefully every day, not every day, but so there are more and more awareness that maybe doodling may help, with a focus and attention. So, kids with ADHD that trying to get unplugged, just like actively prescribe something like art may be a good way for people to get unplugged. There's a lot of interesting sort of relationship or activity that we can prescribe for people with dementia. When I can go on and on, but there's like a case study, especially the UCSF folks like studied some of the frontal temporal dementia can suddenly become creative, like amazingly creative. And perhaps when they lose a lot of function and quality of life elsewhere, they can find more meaning in their daily lives by having them pick up artwork. And also, it reduces the rate of depression, social isolation, and it gives them a lot more like the power of choice and decisions as they lose those powers in their daily activities. And then in the end stage of dementia, when they can barely talk and can express their emotions, perhaps the art is a way for them to open up their emotions. Currently, just want to share, plug in here that, part of what I do outside of Queens, partnering up with an architect, Mari Kim, who's behind me, just joined my office to join this talk, and also Helena from Honolulu Museum of Art. We're trying to create a novel way of bringing art classes to promote brain health, and we're in the middle of brainstorming session, and also a way to create a architectural design to promote brain health. So this is sort of like a outside of the healthcare attempt to tackle brain health in the community. Now I'm going to switch gear and talk about sleep, neuroplasticity and sleep. So sleep is really important to lock in that memory, what you learn during the daytime. So during the daytime, you try new things, neuroplasticity, challenge yourself, fail, correct it, and then try to learn and memorize. And then at nighttime, hopefully that's going to consolidate into more of a permanent memory. So when you wake up, you retain that memory. That's the key, right? And sleep deprivation doesn't help. And also, this is a correlation between the BDNF, the brain fertilizer, and sleep. So if you're sleep deprived, like me last week when I was sleeping like 2 hours a day for 7 days, definitely like I did not have BDNF, and I'm trying to catch up right now. So sleep is really important to produce your own brain fertilizer. There are all these environmental factors that can lead to impaired brain conditions, and one of them is sleep deprivation. Now, changing topic just a little bit, but there's a correlation between sleep and this. I want to introduce this new concept of glymphatic system. This is something that I wasn't taught in medical school or residency. This concept came out maybe like almost like a decade ago. That's relatively new to the neurologist. And we learned that just like the body, which has a lymphatic system, brain has a glymphatic system basically. So brain makes waste, toxic product that we need to clear. How does that do that? It's actually through these small little channels, microscopic channels that we have, and that's how we actually clear them. So there's an inflow of CSF right around the small arteries, like arterioles. Okay, there's a small little channel between the brain interface and here, small little sliver. Okay, CSF runs through there. It's like a little gutter of like water constantly flowing. And then that CSF goes through the brain parenchyma, the aquaporin 4, which is a channel that promotes like water exchange. And so CSF goes through in the brain parenchyma, there's an exchange that happens where all these sort of waste byproduct that our neurons and astrocytes create gets kind of like picked up and then goes through this perivenual space, okay? And then if you have a good working vein, it gets flushed out. Okay, that is the glymphatic system. Now, if you have issues with venous outflow, you may have the waste product like build up. If you have a lot of waste production, that's also a problem. And then again, like any sort of pathway problem here can cause lymphatic system issue. And that is actually one of the reasons why the stroke neurologists are heavily really into dementia and lymphatics right now. At the International Stroke Conference last year when I went, there's a whole section where vascular neurologists were like there talking about lymphatic systems and hypertension, vascular health, and dementia. Now, this is sort of a conceptual model for what can lead to neurodegeneration. It's kind of a busy slide, so I just want to just not go through the whole thing, but I just want to highlight that one of the big things is the lymphatic system impairment. That is one category now we're actually respecting that can potentially cause to problem and sort of a possible dementia. Now, it was super exciting that about 10 years ago in 2013, there was a big news release made a splash, at least within the neurology community, that sleep, okay, during sleep, that's when this flushing happens. It's almost like flushing your toilet. That happens when you're sleeping. That's when you flush out your toxic waste. That kind of makes sense, right? Like when you take a power nap, when you have a good sleep, you feel so refreshed. And if you don't, you feel like your brain's foggy. I mean, I don't think that's the reason, but sleep is super important for this lymphatic system flushing effect. Again, this was the sort of the animal model study that showed that basically around the vessel, these are all vessels, the clearance of all these things that they're highlighting was a lot more robust during sleep. So just sort of have this in mind when you think about how much sleep you want to get every night. Okay, and then also we're understanding more that lack of sleep in middle age may increase the risk of dementia. Now I'm going to switch gear. This is all about lifestyle, so I'm going to keep changing gear. Neuroplasticity and diet and when to eat. We often talk about what to eat, like berry, plant-based diet, whatnot. We often don't talk about when to eat, right? So I want to kind of like focus on that first. So fasting, fasting may not be for everyone, but fasting process has a very, very robust impact in your body and your brain. And it actually changes a lot in terms of sort of energy metabolism. It optimizes the neurons like bioenergetic, the neuronal metabolism, and then optimizes the neuroplasticity and neurogenesis. So fasting improves cognition, stalls age-related cognitive decline, usually slows neurodegeneration, reduces brain damage, enhances functional recovery after stroke, and mitigates the pathological and clinical features of epilepsy and multiple sclerosis in animal models. In epilepsy, when we have refractory seizures, we often do ketogenic diet. So it's kind of like a fasting process. We stop the glucose use and flip them into ketosis, and they often stop seizing. There are different kinds of fasting. I'm not gonna go through which one we should do. I actually don't know which one's better, but they have like a pure fasting where you don't drink or eat anything, which is really hardcore, to like water only fasting, to fluid only fasting, which is coffee's okay, tea's okay, nothing with sugar, and then there's a fasting. There's a limited calorie intake fasting, so you can eat and drink, but keep your calorie down. And there's a different kind of frequency, like you do it like every day for a few, several hours, or you alternate days and so on, and 2 days a week, and occasionally do hardcore. Again, I'm not here to say what works or what's better, but just want to share with you that there are different kinds. Fasting is not for everyone, and I'm actually not sure if it's something that medically advised for elderly patients that you guys take care of, so I don't know. But at least for a middle-aged adult or they're relatively healthy, we can uh, I guess, somewhat recommend trying. However, you want to make sure that they don't have certain medical conditions that may be a contraindication. Um, people of low body weight, which may be for fragile elderly people, pregnant women, extreme age, again, so a very old population may not be advised. And then, type 1 diabetes, renal stones, gout. These are some of those adverse effects. Um, this picture really tells a story. This is thanks I think Brad Wilcox is on the line and he shared a lot of these slides. Caloric restriction has a very powerful anti-aging effect. So these are two monkeys they're not cloned, but they have similar genetic makeup and they're both old and they age similarly. They had the same kind of living environment, same kind of like food diet. The only difference was that mom was given whatever amount of food, unrestricted, eat till you're full, and then the other one had a sort of a restricted amount of food given, restricted calorie. So the one on the left, the one that looks kind of like more gray hair, more wrinkles, more not scoliosis, but curved back, less muscles, less fur. That's the one that was fed whatever, eat till you're full. And then the one on the right, someone that had a moderate caloric restriction. So look how like muscular, more fur, less gray hair, and it is pretty amazing. So caloric restriction sort of kind of shocks your body to say, hey, you may be starving to death. You gotta turn on all sorts of survival gene to help you survive, and that process really is beneficial to the body. Um, and sorry, this may be a little out of sync, but you know, sorry. So that's that for the caloric restriction. Um, to protect and prevent brain aging process and to promote neuroprotection, you got to reduce inflammation that happens in the brain. And there's a whole thing that you can do to tighten the blood-brain barrier so you don't have a lot of these sort of things going into the brain. And there's an epigenetic sort of factor where environment can really trigger what you express. And then the metabolic waste clearance that we talked about. One thing that we are starting to sort of appreciate is the whole gut-brain axis and then the neurodegeneration. So really what we eat is who we are, our brain, really. So we have a lot of intestinal flora that makes a lot of these sort of microbiome metabolites. And also what we eat and what gets digested actually goes into the brain through making various kind of neurotransmitters as well. And also it can promote or reduce the inflammation. And then if you have a leaky gut, you can actually have more inflammation. And then the point here is that caloric restriction can also positively impact this whole process. Now, in that busy slide that I showed you, there's at the top, talked about blood-brain barrier permeability. And then on the right, which is basically like a gut health, um, I just want to sort of zoom in here a little bit and then highlight what caught my attention. So butyrate produced by gut microbiota upregulates tight junction proteins and reduce blood-brain barrier permeability. So butyrate from the gut microbiota tightens the blood-brain barrier. Okay, that's number one. And also that prevents things from going into the brain and makes less likely to have brain inflammation. So the other thing on the bottom is that butyrate produced by gut microbiota attenuates pro-inflammatory cytokine expression in microglia. So what is butyrate? So butyrate is produced when sort of like our good bacteria in our gut, or our gut microbiome, break down fiber in the colon. Okay. We basically digest and absorb everything else, right? Like refined sugar and so on, that. But our body just can't break down fiber during the digestion process. So it's left for gut bacteria to break down. and then butyrate is produced from that process. And that is one of the reasons why high fiber diet is good for the brain. I never understood that till now. Um, so it's also, so it's the fermentable fibers that actually make the butyrate. So what should we eat to increase the butyrate so our brain gets healthier? Well, a lot of the fruits, vegetables, legumes, nuts, seeds, and whole grains. I can actually have a whole list of things, but I just put into the category. But basically, that's the genesis of why plant-based diet is good for the brain. And the other thing is fermented food. So kimchi, natto, sauerkraut, kombucha, yogurt, and a couple other things I probably missed. And then also some things like seaweed, microalgae, and lastly, dark chocolate, which is good for you. So now there's more interest in things that we in the Asian American Asian community we enjoy, such as kimchi, natto, which is fermented soybean, and like something like kombucha. um, Now there's more and more interest in trying to understand the diet that's been working for certain area, like a blue zone place, like Okinawa. Thanks, Brad, for sharing this slide. So this is sort of like a traditional Okinawan diet, and then his group is trying to understand why those food are leading for them to live like long life. So Dr. Brad Wilcox and his brother. They've been together running the Okinawa Centenarian Study for some time and published a bunch of things. And then the sort of the common theme is that these Okinawans who live long are constantly moving, and then they're not eating a lot. They're eating high fiber and then complex carbohydrates, and they're leaner, and they tend to have a caloric restriction phenotype, so they burn more calorie than they consume. And that's one of the key. Now, we're very fortunate that they're part of our sort of a research collaboration. Again, there are several places around the world that are considered blue zones, and Okinawa is one of them. Um, Now, since I have some time, I have about 10 minutes, I would like to show some of the research projects that I've done with Dr. Masaki here. Um, so Honolulu Heart Program is one of the longest running cohort study. It started in the 1960s, and Dr. Masaki oversees that currently. Dr. Brad Wilcox is also part of that. And Dr. Wilcox is was the main PI who discovered the longevity gene, which is called FOXO3 gene. So this is one of the two longevity genes. Maybe there are more now, but at the time it was FOXO3 and ApoE. Those are the only two longevity genes. That's actually as a resilience gene that once it turns on, it hits multiple pathways, and not just one pathway, multiple, and makes you healthier and then age well. And it's been shown that it actually reduces all-cause mortality in different ethnic groups, as well as the Japanese Americans here. And it tends to heavily impact the coronary heart disease mortality compared to cancer and others. So when I joined the group, we want to see if FOXO3 has any relevance in the risk of hypertension. And FOXO3 alone doesn't prevent hypertension. Prevent intracerebral hemorrhage or hemorrhagic stroke. But what we found was that it has a relevance if you have hypertension, meaning that if you don't have the good longevity gene and if you have hypertension, you're more likely to get intracerebral hemorrhage compared to if you're not having hypertension. That's like what we know, right? Like compared to non-hypertensives, if you have hypertension, you're more likely to get hypertensive hemorrhage. Duh, right? But if you have this good longevity gene, we found out that that difference was no longer significant, meaning that if you're lucky to have that gene, and if you have hypertension, you may be lucky that you're not going to get a stroke. Okay, that's kind of what we found. So this is conceptually what we're saying. So if you have hypertension, if you have the regular non-longevity allele, you're going to have a rupture, bleed. But if you have that good FOXO3 or the resilience gene, you may be lucky to keep your blood vessels relatively healthy over time and may not have that bleed. But if you're normotensive, doesn't matter what you have, your vessel is going to stay healthy. Now, that was maybe like 4 years ago, and I started to really think about dementia. I'm a stroke neurologist, I'm a neurointensivist. I don't see dementia patients, I don't even have a clinic, but I start to really think about dementia because we know that dementia is going to hit us all in the next 20, 30 years. It's going to be like the massive new slow pandemic that's going to really impact us. And then the science was of the vascular neurology and dementia neurology were merging more and more now. At the International Stroke Conference, we talked a lot about dementia, and a lot of my colleagues are now talking about dementia prevention and not stroke prevention. And that's because hypertension matters. So the more hypertensive you are, more likely you're going to get dementia. Whether it's vascular dementia, Alzheimer's dementia, it doesn't matter. Hypertension increases the risk of Alzheimer's dementia. So what does hypertension do to your blood vessels in the brain? Well, it kind of impacts the small vessels, okay? And it can cause basically lacunar infarct, small vessel disease, and also microinfarcts. And microinfarct is something that I got interested in. So what is cerebral microinfarct, which is an invisible, small little microscopic infarct that you can only appreciate in a neuropath study. You can't see it on the MRI. It's only when you have an autopsy or brain biopsy, you can actually pick that up. And there are not that many studies with a lot of autopsy or brain biopsy samples. And luckily, the Honolulu Heart Program from Kokini has that information. So we thought this was a great opportunity to better understand this and how FOXO3 plays a role. So this is one of the hypotheses as to why microinfarct may increase the risk of dementia. So it's not just by losing the brain cell. That's what I thought. Basically, I thought maybe if you're missing or losing a couple neurons here and there, that's how you get dementia. But that may not be the whole story. So when you have microscopic cerebral microinfarcts, you may actually cause sluggish glymphatic drainage system. And then basically the CMI makes the sluggish lymphatics, which increases the likelihood of all these amyloid and other waste product to accumulate in the brain. There's one study that had the brain autopsy samples, and that showed that microinfarcts were associated with dementia. So our research question was, can that associated FOXO3 gene, the good gene, impact this sort of a relationship between hypertension and the risk of cerebral microinfarct? So our hypothesis was that if you have this common regular FOXO3 genotype, not the longevity one, okay, if you don't have hypertension, it's fine. Your brain's gonna stay healthy. But if you have hypertension, you can have a bunch of cerebral microinfarcts, okay? And if you have the lucky longevity gene, whether you are normotensive or hypertensive, you're lucky. Your brain is going to stay healthy. So that was our working hypothesis. So we had about a little over 800 men with brain autopsy data. This was done back then in a systematic fashion. And then we actually basically dichotomized the outcome variable into having any cerebral microinfarct, yes or no. So whether you had 1 CMI or 20 CMI, that was counted as yes, there was any microinfarct. It was just too complex to do some kind of analysis where you had to quantify how many microinfarcts. So yes or no, yes or no. And basically, if you look at the group who had no cerebral microinfarct, less likely to have hypertension. That's kind of intuitive, and a little bit lower BMI and a little bit lower fasting glucose, or very modest difference. And then, lower rate of a stroke. And in the multivariable model, it was kind of depressing initially because only age, BMI, and glucose came out as the independent predictors of cerebral microinfarct, somehow hypertension was no longer a significant predictor of cerebral microinfarct. And I was just shocked. I was shocked that it was not. I just thought that hypertension was the main driver for these small little microinfarcts. So instead of saying, well, the project is over, we're done, we did an interaction analysis. It turns out that there's a significant interaction between FOXO3 gene, so the longevity gene, and hypertension in this relationship. And what does it mean in sort of like a simplified version? Well, it means that if you don't have that longevity FOXO3 gene, hypertension was a robust predictor of cerebral microinfarct. So odds ratio of 2.1, hypertension, very important for microinfarct formation with a p-value 0.04. But if you had that longevity gene, it didn't matter. Hypertension was no longer a factor for that microinfarct. So again, we concluded that this hypothesis may be true, that if you don't have that FOXO3, hypertension drives that microinfarct. And if you have that longevity gene, it protects the bad effect of hypertension on the blood vessels in the brain. I just want to thank all the members of the team who helped me with this project, especially Dr. Masaki and Dr. Wilcox, who joined us today. And that's all I have. Thank you, everyone. I'll stop sharing. I see a lot.
Guys, thank you so much for a wonderful presentation. We'll open it up for questions. And since we have a relatively small group, please unmute yourself to ask Kaz the question. Or you can type it in the chat if you like.
Hey, Kaz, this is Abe. Thank you very much. That was a great presentation. I think you're absolutely right that treatment for Alzheimer's disease is going to be limited if we wait until the disease has progressed to a more severe state. And so preventative approach makes much more sense. And I really like the idea of using exercise, because exercise is really good for you in a lot of ways. But based on your research, do you have a sense of whether it's the aerobic exercises or more like weightlifting kind of exercises? Does it matter? What do you recommend?
Yeah, that's a great question. To be honest, I don't know the answer. I'm getting a sense it's the high intensity aerobic exercise more than like weightlifting, but I'm not sure.
Great, thank you.
Ilan, would you like to unmute yourself?
Hi, thanks. Can you hear me okay?
Yes.
Yeah, I was just curious, Kaz, if you could speak to biomarkers that are available that are relevant to brain health. BDNF has been looked at, I know, both in the CSF and peripherally. And gosh, it would be great to have something like a serum biomarker that could sort of speak to what processes are going on in there that doesn't require a multi or tens of thousands of dollars worth of imaging and whatnot. Do you know where the state of the science is with regard to that?
Yeah, I think Abe can probably answer that better than I can. I know like a blood serum, like tau amyloid, something is kind of like on the horizon. I'll probably defer to Abe.
Well, the biomarkers for diagnosing Alzheimer's disease, they're making headway, but for brain health, that's kind of beyond me. And so I'm not quite sure what to look for in someone who is preclinical in terms of memory impairment. But there are certainly CSF evaluation or PET scan with amyloid staining to make diagnosis of Alzheimer's disease. And there's a new biomarker test on blood that became available for Alzheimer's disease, which is currently being looked at in terms of how useful it's going to be in the clinical setting.
Abe, that blood test isn't FDA approved yet, correct?
Correct. And it's not being covered by any of the insurances right now, but Mayo Clinic has made that available. It's on the market, so you can order it. But like I said, it's not going to replace the CSF evaluation for, say, determining if someone is eligible for lecanemab or other monoclonal antibody infusion therapy. And so the exact role that it's going to play in the clinical practice, it's being worked out right now.
Thank you. Brad, you want to unmute yourself and ask your question?
Sure. Hi, guys. Great presentation, Kaz. Loved it. Sorry, I got a raspy voice. I was just coming back from a short trip. Didn't get much sleep last night, but there's a lot of genetic testing out there for the general public. It's been around for quite a while. And so you can, I think most of us know, you can find out your FOXO3 genotype and your ApoE genotype. And but I think a lot of how shall I say, non-medical folks might like get shocked if they have one of the like E4 alleles as a risk allele or don't have the longevity FOXO3 version. So what do you think regarding the risks and the benefits of doing your own testing?
Are you asking me whether people should get tested or not?
Yeah, well, if they want to. I'm asking you, do you think it's going to help them or harm them?
I mean, okay, I think that's a very deep philosophical question. It's hard to say yes or no, like we want one statement. But I think with an informed discussion, I think there is some benefit potentially if they're aware of the implication. Here's the good thing, there's a paper coming out from Mass General Group that I've been collaborating have shown that. So I should have shown this in this slide, but they developed the brain care score. So all these risk factors, and if you modify and others that predict the dementia risk and so on, they have shown that even if you have ApoE4, if you actually improve your lifestyle scores, your risk goes down. So that conclusion is that it's not hopeless. Even if you have ApoE4, you can actually make a dent if you do it. So if you're sort of if you have a positive outlook, and you can still make a difference, then you want to get it tested so you can start early. It's one thing if you can't do anything about it, you have ApoE4 and you're doomed, whether you do anything or not, then I'm not sure if you want to get tested. But it looks like even with ApoE4, you can do something about it, and you're empowered to do it. It's not waiting for the medication, but it's the lifestyle changes.
That's great. Great news, Kaz. Are you able to share with us a link or something? Or is it just available online? You can just Google it, Brain Score, Mass General.
Yeah, Brain Care Score is published. That new one with ApoE, that may not be in print yet.
Oh, okay. Thank you.
Catherine has a question in the chat. Would you mind unmuting yourself and asking your question?
Yes. As I was looking at the slides and listening, I know or sounds familiar to what Dr. Bredesen is writing in his book, End of Alzheimer's, which I believe people have panned, but I'm reading it because I have a friend who is starting that program, Recode. But I just wondered if you've read it and what you think of it. And because a lot of the processes that you described are written in his book.
I'll be honest, I've heard of his name. I have not read the book yet. But I know that the concept of really focusing on lifestyle changes have a very robust impact on Alzheimer's disease course and sort of risk factor, and even like their outcome is being more and more appreciated by the scientific community. So, but that's your question. I have not read it, so I can't really say much about it.
Okay. Thanks.
I'll just say a couple of things about Dr. Bredesen. He's actually a well-respected dementia researcher who trained at UCSF and held a position at UCLA before he resigned from there. And he's taking some scientifically accepted concepts about what might help improve memory function and is drawing a conclusion that you can use it to treat Alzheimer's disease and the effect of the Alzheimer's disease. And that's the big leap that a lot of the clinical doctors have issues with. And so if you're talking about using those concepts as a preventative approach and sharing the information with the community so they can work on brain health and prevent development of Alzheimer's disease later, that is well accepted. But he's taken those concepts and he's offering these protocols that people can sign up for and pay a lot of money to get the protocol, promising that this might help treat their Alzheimer's disease. And that is not accepted by the scientific and medical community. And that's what we have issues with. His protocol is being franchised, though, because beyond his clinic where he operates in private practice now, there are other people that are buying into his protocols and then making it available to patients at the high cost that he's requesting. And I imagine there's some kickback going back to Dr. Bredesen. I have concerns about that type of an approach. I mean, a good medical foundation for knowledge should be shared without an expectation of profit. And it should be available at any clinic throughout the country for people who have memory impairment to take advantage of it.
Thanks, Abe, for saying that. And I agree with Abe. And I mean, I am aware of that protocol. And my other head of the Brain Health Applied Research Institute is trying to do a similar thing, but doing it carefully, as Abe stated. You know, what we're trying to do is promote all these lifestyle sort of impact on brain, but never promising to cure Alzheimer's or reverse the course. But, you know, sharing this kind of information to say why exercise increase the BDNF, help clear the amyloid plaque, you know, helps with lymphatics, sleep, you know, all these things that are like true, right? I can say that you do this, you may have Alzheimer's, you're going to improve all these other sort of pathways. That's all we can say. And I think that is to me, okay way of approaching. Anyway, I have Stephanie Liu has her hand up.
Yeah.
Hi, I'm Stephanie. I'm one of the Jerry Fellows this year. So I get asked a lot, and I see a lot of like, you know, people's medications, their lists showing like a lot of these supplements that people are taking to, you know, help like just with general health. And there's a lot of marketed supplements that supposedly help with brain health, neuroplasticity, and all this stuff. Do you have any kind of recommendations, or what is your kind of thoughts on all these supplements that are kind of on the market, and if anything has worked or if anything actually has benefit?
I personally don't endorse or strongly recommend any of them. One, because nothing has been panned out scientifically, so I don't feel comfortable endorsing like that. So personally, I just recommend exercise, sleep, art, meditate.
Thank
And
you,
for what
Carl.
it's worth, I'm in complete
Some of them
agreement with that
have
approach too.
some of them have side effects as well. So yeah, sorry, Abe, please jump in.
Oh, yes, I was just going to say that I'm in complete agreement with Kaz's approach to the situation. There's so many people who are worried about memory problems these days. Some companies have recognized that there is a potential for profit here. And so they are asking for a lot of money for these various products that they market heavily, but they really haven't been proven to be of any benefit. And as Kamal just mentioned, for some of them, there can be some risks of using them that we just don't fully understand yet. And so I advise my patients, against using those products that don't require prescription, those that are not FDA approved. I would just stick with the standard medical approaches. And then in addition to that, take advantage of the preventative approaches that were brought up in this wonderful talk today.
Thanks, Abe. Actually, the NIH has realized that so many people are taking these so-called nutritional supplements or nutraceuticals, whatever you want to call them, without their doctor's knowledge. And so several years ago, they actually started a National Institute of Alternative and Complementary Medicine to study these drugs. Because I think part of the problem is a lot of claims are made, but there aren't very good studies on them. So hopefully they'll be studied more so that people don't just waste money and actually take harmful things. Any last questions? We have about 2 minutes left. If not, there's an evaluation link in the chat. So please follow that link and complete your evaluation to get your CME credits. And please give us your comments. We really appreciate that. I really want to thank Dr. Nakagawa for a wonderful presentation. Also wanted to thank Abe for giving his comments. And just to let everybody know, Abe is going to be our speaker next month. So the month of October, Dr. Abe Chung will be speaking for us. Thank you again, Kaz. We really appreciate you doing this.
Thanks, Kamal. Thanks, everyone.
Take care.
Take
Bye-bye.
care.